Okay, good morning. In today's lecture, we're going to be talking about receptors of neurotransmitters. So we're going to be looking at specific receptors for neurotransmitters and some other signaling molecules, but mostly neurotransmitters. My assumption is that you already know quite a lot about signaling cascades, but we'll see during the lecture whether you know the details that I think are important. So if we see that that doesn't quite work, I'll try to explain a little bit more, but we'll see. Why I'm saying it, because in the when I was lecturing to the Czech students, I saw that actually, yeah, the knowledge of signaling cascades is not as good as I thought, but we'll see. One thing to keep in mind when talking about receptors, signaling molecules, effects, etc., is that, first of all, many signaling molecules have many different receptors, which are coupled with many different signaling cascades. So one molecule can have different effects on different receptors, okay? One important thing. The other important thing is those different receptor subtypes can be expressed in different tissues, in different ratios, in different tissues. So one signaling molecule can have widely different effects in different tissues, okay? So those are the things that are good to be kept in mind, because we'll see that as we talk about the individual receptors and as we talk about the effects of signaling molecules, these factors play a role there. Okay, so today I'll mention the types and subtypes of receptors, their signaling cascades, a little bit about where they're expressed, which tissues and which organs have them, and finally we'll talk a little bit about their effects, what they actually do to the cell, what they do physiologically, etc., etc. Okay? Yeah, good. So we'll start with adrenergic receptors, receptors for adrenaline and noradrenaline. Okay, what signaling cascades do adrenergic receptors trigger? Sure, that's the effect. Glycogen breakdown is the effect of activation some of the receptors. Okay, all adrenergic receptors are G protein coupled receptors. Okay, all of them, no exception. Okay, so that's one thing to remember definitely. All adrenergic receptors, G protein coupled receptors. Now, we have several different subtypes of adrenergic receptors. Do you know what subtypes we have? Okay, we have alpha and beta receptors. But as usual in biochemistry and in medicine, that would be too easy. So we have alpha 1, alpha 2, beta 1, beta 2, and beta 3 receptors. Okay, we're gonna go into a fair bit of detail with adrenergic receptors because they are very important for clinical medicine, they're very important for pharmacology. So knowing about these subtypes is fairly important, I think. You'll see later on that with some of the other signaling molecules, some of the other neurotransmitters, we won't go into such detail. So now there's gonna be a lot of information, but it's gonna get easier and easier as we go further in the lecture. Right, so we have 5 different subtypes of adrenergic receptors. What are the natural agonists on these receptors? Adrenaline and noradrenaline, okay? They're adrenergic receptors. The natural agonists on those receptors, well, are adrenaline and noradrenaline, okay? I will just say now that the two molecules, adrenaline and noradrenaline, bind with different affinities at different receptors. Okay, so some of these receptors have higher affinity for adrenaline, for example, beta 2. Some of these receptors have higher affinity for noradrenaline, for example, alpha-1 or beta-1. Okay? So the thing that I want you to remember is there are different affinities for these two molecules. Again, it's fairly important in clinical medicine because sometimes for some things you use adrenaline, for some things you use noradrenaline. Okay? Right. Let's now talk about the specific G proteins that these receptors are coupled with. So all of them are G protein coupled receptors, but they're coupled with different G proteins. Different G proteins. Right. Alpha-1 receptor is coupled with a G protein called GQ. Now, what does GQ protein do?
Increasing calcium levels.
Okay, how does that work?
Maybe the IP3 that goes to the ER and then releases.
Very good. So there's increase in calcium through IP3 mediated release from endoplasmic reticulum and all other things. But there's one more step that we're missing. Okay, so GQ protein activates phospholipase C, which hydrolyzes phosphatidyl inositol bisphosphate into inositol trisphosphate, diacylglycerol. And these, well, mostly IP3 increases calcium concentration in the cell, and they also activate protein kinase C. Okay? So that's the signaling cascade of GQ receptor, or GQ protein, sorry. Right. Alpha-1 receptors are expressed all over the place, in the brain and elsewhere. But for pharmacology, for clinical medicine, one place is very important, and that's the smooth muscles of blood vessels. Okay? So in blood vessels, there's smooth muscles that control the diameter of the blood vessel, and these express alpha-1 receptors. What do you think will happen if we activate alpha-1 receptors?
Constriction.
Okay, vasoconstriction. The diameter of the blood vessels will get smaller, and what does it mean?
Blood pressure increases.
Indeed. Okay, and there will be an increase in blood pressure. Yeah, as we constrict blood vessels, blood pressure increases. So the activation of alpha-1 receptors causes an increase in blood pressure through vasoconstriction. Okay, that's the function. Well, one of the functions. Anyway. Do you have any questions? No? Let's move to alpha-2 receptors. Alpha-2 receptors are coupled with GI protein. What is the signaling cascade here? What about it? Okay, so GI protein decreases the activity of adenylate cyclase. And because adenylate cyclase synthesizes cyclic AMP, we'll get a decrease in CAMP. Okay? Do you know, by the way, what the I stands for in GI? Very good, okay. Yeah, it's an inhibitory. The S in GS is stimulatory. Good. The Q in GQ, as far as I know, doesn't stand for anything. It's just another letter, but anyway. Alpha-2 receptors are found mostly in the brain. Well, in the nervous tissue anyway. And they are a special kind of receptors. They are presynaptic receptors. Now, we talked about presynaptic receptors a little bit in one of the previous lectures. What do they do? What is the role of presynaptic receptors? Okay, so they're there to mediate feedback to the presynaptic neuron about the amount of neurotransmitter that's been released into the synapse, into the synaptic cleft. Okay, so alpha-2 receptors are presynaptic receptors on adrenergic synapses. What will happen if we activate alpha-2 receptors? What will happen in the synapse?
Inhibition of 7 molecules.
Inhibition of?
Signaling.
Yes. So the release of adrenaline and noradrenaline on the synapse will be inhibited. There will be less adrenaline or noradrenaline released into the synapse. So it's a feedback inhibition. Okay? There's a lot of noradrenaline in the synapse. It binds to alpha-2 receptors and signals to the presynaptic neuron, stop releasing that much noradrenaline. Okay? It's a feedback inhibition mediated by alpha-2 receptors. Yes?
Does this receptor inhibit both like releasing of adrenaline and noradrenaline, or just adrenaline or noradrenaline?
Both. Let's say both. Okay? Both of them bind to alpha-2 receptors.
And their release is only controlled by CAMP pathways?
No, their release is not controlled just by CAMP. Actually, as we all know, the release of neurotransmitters is mostly regulated by calcium. Okay, so calcium is the signal. CAMP via PKA modulates the activity of calcium channels, of sodium channels, of all sorts of other things. Okay, so the CAMP signaling system modulates how much calcium gets in, how much, etc., etc.
It is at the start of the cascade.
It's
CAMP. It is at the start of the signaling cascade that then goes to calcium.
No, it's not. Because as you'll recall, the increase in calcium in the synapse is caused by membrane depolarization. So CAMP normally, in the synapse at least, has no effect. I mean, we don't need it. But it's there, and it modulates what happens with this calcium, with sodium, how easy it is to open sodium channels, how easy it is to open calcium channels, etc., etc. Okay? So it is just something affecting the main signaling mechanism. Okay? Right. So alpha-2 receptors are presynaptic receptors, fairly important in pharmacology, and that's how they work. So they're inhibitory. They're an inhibitory receptor. They're inhibitory receptors on the presynaptic neuron. Right. So those were alpha receptors. Now, let's now have a look at beta receptors. Here, the signaling cascade is easier because it's the same for all three of them. All three beta receptors are coupled with GS proteins. So what's the cascade there? Okay, adenylate cyclase is stimulated. We get an increase in CAMP, and then all sorts of other things happen, PKA, and etc., etc. Right. Where are these receptors? What do they do? Beta 1 receptors are mostly found. Let me just. These three receptors are found in the brain. Okay. So these three receptors, we can find them in the central nervous system. Okay. But apart from that, beta 1 receptors are found in the heart. Okay. So these are very important receptors regulating the heart, and specifically, they have an effect on heart rate and on the strength of contraction. Yes?
I'm sorry, was that B1 or all the beta receptors?
Just beta 1. Okay? So the only thing that's common is the signaling cascade. Okay? Otherwise, I'm going to talk about each of them individually. Okay? So beta 1, important for the control of heart rate and also the strength of contraction, how strongly the heart contracts. Okay? So the activation of beta 1 receptors will increase the heart rate. So that's why adrenaline and noradrenaline increase heart rate when they're released into the bloodstream. Okay? Via the action on beta 1 receptors. Beta 2 receptors. Yes. Beta 2 receptors are expressed, again, all sorts of places, but they're also expressed on the smooth muscle molecules in the bronchi, in the lungs, but also in blood vessels. Okay? Smooth muscles in blood vessels, smooth muscles in the bronchial tree. Okay, do you all know what bronchi are? Yeah, good, yeah. So there we find beta 2 receptors. In the smooth muscle, if you have a question, ask anytime. No? Okay. In a smooth muscle cell, the activation of GS, adenylate cyclase, CAMP, does not cause contraction, even though it's a stimulatory G protein. It actually relaxes the smooth muscle. Okay? Via phosphorylation of myosin light chains, etc., etc. Okay? So the activation of this in a smooth muscle cell relaxes the smooth muscle. So in the bronchi, the relaxation will increase the diameter so that we can get more air into the lungs. Okay? That's what beta 2 receptors, the activation of beta 2 receptors does in our lungs. In the blood vessels, it dilates blood vessels. Okay? Beta 2 receptors dilate blood vessels. Now, maybe some of you are thinking, that doesn't make any sense, because adrenaline and noradrenaline, they bind to both alpha-1 receptors that contract blood vessels and beta 2 receptors that dilate blood vessels. So how does it work? I mean, surely the blood vessels can't know what they should do. If our adrenal medulla releases lots of adrenaline, the blood vessels should be just confused. So how does that work?
Different affinity.
There's a different affinity. Very good.
Do you mean why or how? Yeah.
Either.
To increase the oxygen uptake in the lungs.
Okay. So the reason why we need oh, you mean with the bronchi? Yeah. That's correct. So we dilate bronchi because we need to get more oxygen into the lungs. But now we're talking about blood vessels.
Yeah, but you also dilate the blood vessels in the lungs to have more alpha layer.
Okay.
I guess what you want to say. Yeah.
Oh, okay. All right. Okay, so that's correct. Yeah.
To get more blood to the vessels, so they can get
Very good. Very good. So this may be confusing thing, but I see that none of you are confused because you already know the answer. Is first of all, there's a different affinity for adrenaline and noradrenaline, so there will be different activation based on whether adrenaline is released or noradrenaline is released. But most importantly, different blood vessels express different receptors. Okay? So in a situation where adrenaline is released, in a stress situation, for example, you know, if a saber-toothed tiger is, you know, running after us or something like that, and we have to run away, not to get eaten, some of our blood vessels need to be dilated in order to provide as much blood as possible, for example, muscles or lungs. But some other blood vessels can be constricted, well, should be actually, so that some of the organs don't use up all the oxygen that we need for running. For example, in the gut. We don't really need a lot of blood in the gut when we're running. We don't need to be digesting things. Okay? So in a stress reaction, even though it's mediated by one type of ligand, by adrenaline and noradrenaline, okay, it actually has different effects in different tissues because that's what we need. We need to dilate some blood vessels, but we need to constrict some other ones. And that's mediated by two different adrenergic receptors, alpha-1 and beta-2, and it's possible because different blood vessels, or the smooth muscles in different blood vessels, express different receptors. Okay, does it make sense?
Which receptors did you say? Alpha-1?
Alpha-1 and beta-2. Yeah, it would be easier if it were alpha-1 and beta-1, wouldn't it? But yeah, it just isn't, yeah, unfortunately. Okay, the last subtype of adrenergic receptors is beta-3 receptor. And beta-3 receptor is expressed primarily in the adipose tissue, in the fat. What do you think will be the effect there of the activation? Lipolysis, exactly. Lipolysis, beta-3 receptors mediate the stimulation of lipolysis by adrenaline. Okay? Again, we can think about it as part of the stress reaction. We need to provide energy for the muscles to be able to run away from the tiger or whoever. Okay? So releasing some fatty acids from the adipose tissue is actually very useful, because then they can be burned in the muscle. Okay?
Sorry, beta-3 again?
The what?
Beta-3.
Yeah, beta-3 receptors are expressed on the adipose tissue, and their activation increases lipolysis, the degradation of lipids, of triacylglycerols into fatty acids and glycerol. And fatty acids are released into the bloodstream so that other tissues can use them. Okay? Beta-3 receptors also, and you don't have to remember that, also activate the brown adipose tissue. The brown adipose tissue, of which you've heard before, I'm sure, is used to provide heat in some animals, maybe in humans as well. And beta-3 receptors activate that as well. But clinically, it's not very important. Obviously, pharmacologically, there have been attempts to use beta-3 agonists to treat obesity, but so far, not very successfully. So. One last thing, I mentioned that the first 3 subtypes are found in the brain. Now, where in the brain? Remember when we talked about neurotransmitter metabolism, I said that by far the most important neurotransmitters are the most abundant, not important, the most abundant neurotransmitters are glutamate and GABA. Okay? All the other interesting ones are just minority neurotransmitters. So if we want to see where in the brain we have noradrenaline or adrenergic neurons. Actually, there's just a relatively small group of neurons in the medulla oblongata. I know you don't know the anatomy of the brain, so this is just for fun, okay? You don't have to know that. There's a relatively small group of neurons that produce noradrenaline or adrenaline in the brain, but they project, their axons project to most of the brain. And their role is that they have 2 main roles. One is they regulate mood, so they play they play a role in regulating mood. Okay, if we're happy, if we're sad, okay. That's what noradrenergic neurons do, at least partly, and we'll talk a little bit more about that in the second pharmacology lecture. But they also activate arousal, whether we are conscious or not conscious. Very important, whether we go to sleep or whether we're active. That's regulated by noradrenergic neurons. Okay? Very important. It's part of the reticular formation, if anybody's heard of it. Anyway, any questions about adrenergic receptors? If not, let's move on to dopamine, dopaminergic receptors. Now, by all means, take notes. That's very good. But I'll give you, I'll post on the VUCA database a table with all the receptors that you need to know, with all the signaling cascades that you need to know, with all the agonists, antagonists, effects, and everything. Okay, so it's going to be a table summarizing everything. Do take notes; they're very useful. But there will be a summary table with all this information. Okay. At least most of this information, maybe not all of it. Okay, dopaminergic receptors. Similar to adrenergic receptors, there are many subtypes. Currently, there are at least 5 subtypes of dopaminergic receptors, but we'll talk in more detail just about 2 of them. I'll just put down. Okay, so at least 5 subtypes are known. The signaling cascades triggered, so we're going to talk about D1 and D2, okay? The signaling cascades triggered by D1 and D2 are GS and GI. Okay, again, you know the cascades, so what happens there? So D1 receptor is mostly stimulatory. D2 receptor is mostly inhibitory. Okay, but it also differs depending on specific tissues. Dopaminergic receptors, again, are expressed in many tissues in the body, but pharmacologically and clinically, we're mostly interested in dopaminergic receptors in the brain. Those are the ones that we're really interested in. Okay, so we're only going to be talking about the effects in the brain. Okay. As I said with noradrenaline or noradrenergic neurons, dopaminergic neurons are also formed just two very small groups of neurons somewhere around here. Okay. Relatively small nuclei. I'll give you the names of them. You don't have to remember them. Just, next year when you learn about the structure of the brain, they'll ring a bell. One is called the ventral tegmental area. And the other one, which you may have heard about, is called substantia nigra, the black substance. Okay, so these are the 2 dopaminergic nuclei in the brain. Once again, from there, the axons are sent all over the brain. There are 4 main projections. This is just for information, okay? You don't have to remember that. There are 4 main projections of dopaminergic neurons in the brain that serve different functions in the brain, okay? One of them is the projection from ventral tegmental area into the cortex, okay? This projection is important for regulating thinking. Okay, now I put it in inverted commas because what does it mean, thinking? This projection has been implicated as part of the problem in schizophrenia. So in psychosis, when thinking breaks down, the projection from VTA into the cortex plays a very important role. And we'll talk about that more in the second pharmacology lecture, okay? So just now remember there's something with dopamine and regulating proper thinking in the brain. The other important projection of dopaminergic neurons is from substantia nigra into something called basal ganglia, somewhere around here. That is crucial for regulating proper movement. And it's damaged in which disease? In Parkinson's disease. Okay? Parkinson's disease is basically the damage to these dopaminergic neurons in substantia nigra, and therefore a problem with this projection into basal ganglia. Okay? So that's the second projection involved in motor coordination and regulating proper movements. Another very interesting projection of dopaminergic neurons, and again we'll mention a bit more in the second pharmacology lecture, is from the ventral tegmental area into, one of the basal ganglia, part of the limbic system. I know it doesn't mean anything to you now. But this projection is part of what is called in neuroscience a reward system. The brain reward system basically gives us a reward for things that we're biologically programmed to do. Okay, it's a motivation. It's a motivation circuit to motivate us to do things like eat chocolate, have sex, you know, other things that people might be motivated to do. So this crucial projection from this one very small group of neurons into this other very small group of neurons is a very important projection. And in pharmacology, especially the pharmacology of drugs, of illicit drugs, this circuit plays a huge role because many drugs that are abused activate this reward circuit, and that's why people become addicted to them. Okay? So that's a dopaminergic system, a very important one. The final, fourth dopaminergic circuit that's quite important, but we will not talk about it very much, is a circuit whereby dopaminergic neurons regulate the production of prolactin. You've heard of prolactin? Okay, so the production of prolactin is regulated by dopamine, which is only important, well, it's important for all sorts of things, but it's also important because some of the drugs that inhibit dopamine will increase the production of prolactin, which actually causes very unpleasant side effects, especially in men. It's actually quite a serious matter. Right. Those are the dopaminergic receptors, or at least the ones that we're going to be talking about. From a pharmacological point of view, D2 receptors, very important. Again, we'll talk about them later. Any questions about dopamine?
What did you say about prolactin? The production of prolactin?
The production of prolactin is inhibited by dopamine. Okay, so there are neurons that inhibit the production of prolactin. If those neurons are inactive, or if the dopaminergic pathway is inhibited, prolactin production will go up. Okay? Yep.
Something you said there a second about adverse side effects or bad side effects.
Yeah, I'll talk about them a bit more in the lecture later on, but that's the problem. So you have drugs that inhibit dopamine for some other reason, but at the same time they will increase prolactin production, which is something you don't want usually. Okay. Any other question? No. Okay, let's move on to serotonin. The abbreviation for serotonin usually used in literature is 5-HT, and indeed the receptors are called 5-HT and a number. Now, the latest database of receptors shows at least 6 subtypes of serotonergic receptors, and to make it even more fun, a lot of them have actually subtypes. So you get receptors like 5-HT1A, 5-HT2C, stuff like that. I will only talk in a bit more detail about the first 3 of the family, so 5-HT1, 5-HT2, and 5-HT3. Unlike dopamine receptors, and actually adrenergic receptors, which were all coupled with G proteins, 5-HT3 receptors are an ion channel, a ligand-gated ion channel, a cation channel. Okay, so 5-HT3 is a receptor that's completely unrelated, chemically, structurally, is completely unrelated to all the other serotonergic receptors, but they all react to the same molecule. But this is a cation channel. These two, and actually all the other ones, are G protein coupled receptors. Okay? And the G proteins they're coupled with are GI and GQ, with the corresponding cascades. Now, what is the function of the serotonergic system? again, there are several different functions. The serotonergic nuclei. Do you know what a nucleus is when talking about the brain? Yeah? So it's a group of neuron bodies, of cell bodies. That's called a nucleus. Okay? So in the brain, without trying to go into much detail, well, you have gray matter and you have white matter. Okay? The gray matter you find in the cortex. But when you find bits of gray matter elsewhere than in the cortex, they're usually called nucleus. Okay? So it's bits of gray matter elsewhere than in the cortex. That's a nucleus. Okay? So the serotonergic nuclei are found basically somewhere like that. Okay? For those who are really interested, they're called nuclei raphe, whatever. You don't need to know that. And as usual, they project all over the brain. Okay, into the midbrain, into the hypothalamus, into the cortex, everywhere. What is their function? The basic three functions of serotonergic systems in the brain. One is very, very important for pharmacology, and that's the regulation of mood. Okay? Very important, connected to depression. We're going to talk about that later on. The other important role, even though much less studied or much less understood, is regulation of perception, something with perception. And how do we know that serotonin plays a role there? Because certain drugs that affect the serotonergic system cause hallucinations. Okay, again, we'll mention more details about later. But serotonergic system does seem to play some role into integrating perceptions and making sense of what we see and what we hear. Okay? If that's dysregulated, we may see or hear things that actually aren't there. Yeah?
What was the first?
Mood regulation. Okay? So sadness, happiness. Okay? Mood. With the hallucinations, it's the 5-HT2 receptor that's mostly connected. With the mood, it's most of them, it seems. And the final function that I want to be talking about now of serotonergic system is mainly mediated by 5-HT3 receptors. And that's the control of vomiting. Again, pharmacologically very important. Our body can be triggered to vomit by all sorts of stimuli. Okay, you all know that stimulation of the throat from the inside will cause vomiting. There may be vomiting caused by something going on in the stomach, but there's also a mechanism by which the brain monitors the composition of the blood, and if it finds that there is some toxic substance in the blood, it triggers vomiting. Okay? The idea being, you know, if you eat something bad, the brain finds out that there's something bad in the blood. It tries to get rid of whatever remains in the stomach so that you don't die of the poisoning. Okay? There's only a relatively limited number of chemicals that will trigger this mechanism, but it's a very important mechanism. Okay? So the brain monitors the composition of the blood. If there is some toxic substance, certain types of toxic substances, it will trigger vomiting. The vomiting there is important because it's very difficult to stop. As long as the brain feels that there's something wrong there, you will keep vomiting. Now, why is it important in clinical medicine? In chemotherapy, most of the substances that are given are the ones, or many of the substances that are given are the ones that trigger this kind of vomiting. And before there were drugs that could prevent that, the vomiting was the main negative effect that would basically stop chemotherapy. You would have to stop chemotherapy because the vomiting was just so intractable, so impossible to do anything with it. They had to stop giving chemotherapy, which obviously wasn't very good for the patient. This kind of vomiting is mediated by 5-HT3 receptors, and we can stop it by targeting some of the drugs at 5-HT3 receptors. Okay, again, we'll talk more about that later on. Okay. Any questions?
The third one was their perception.
Well, the first one was mood. The second one was something with visual and auditory perception and integrating it with other things. That's kind of a nebulous, not well understood mechanism. And the final one that I want you to know is vomiting, a specific type of vomiting. Yeah?
So those drugs, will they inhibit nausea as well, or just the reflex vomiting?
They will inhibit nausea caused by toxic substances in the blood. Okay? 5-HT3. They won't have any effect on kinetosis. So if you're sick in a car, these have no effect. Okay?
But you were also saying that all of them were kind of regulating as well.
Probably not so much 5-HT3. Okay? These seem to have very specific function in the brain. Okay, any questions about serotonin?
What about HT1?
Sorry, I can't even see who's talking. Can you raise your hand? Oh, sorry. Yeah. Okay.
What about HT1?
5-HT1. They are involved both in the mood regulation. They're probably involved in many of the functions of serotonin. Again, 5-HT3 and vomiting, that's a very clear connection there. The other ones, they all probably do something, okay, in all these functions. Why I pointed out 5-HT2, because some of the interesting drugs work on 5-HT2, okay? But otherwise you can't say specifically this does this and this does that, okay? There are no more, yes?
Just a quick question. SSRIs, they mainly target...
We'll talk about them in the second pharmacology lecture, okay? Right, there are no more, yeah?
Remember that reward system you were talking about like a second ago? Like
Yeah, yeah, yeah. Yeah. Okay, so the reward system is a dopaminergic system. So now we talk about serotonin.
Yeah, I know.
But yeah, that's fine. So it's a dopaminergic neural circuit that gives us motivation for certain biological things. Okay, so it rewards us for doing the things that we were evolved to do. Okay. Okay, let's have a 5-minute break then. Let's carry on. Do you have any questions about the first half? No? Okay. Because there are loads of questions during the break, so, Okay, no more questions now. Good. So moving on to the next neurotransmitter, that's going to be a super minor neurotransmitter, but an important molecule in pharmacology and in clinical medicine, and that's histamine. Now, there are at least 3 histamine receptors, histaminergic receptors, but we'll just talk about the first 2, H1 and H2. They are both connected with G proteins, and namely they are GQ and GI. Okay, again, the cascades, So, yeah, those are the G proteins. Now, what about the function? H1 receptor is responsible for mediating, well, physiologically for mediating inflammation. Okay, at least part of inflammation. Okay, there are other mediators of inflammation, but histamine is one of the mediators of inflammation. So the redness and the swelling of an inflamed piece of tissue is at least partly mediated by H1 receptors. Pharmacologically, the more important function of H1 receptors is mediating allergic reactions. Okay, so allergic reaction like swelling of the nasal mucosa, the production of liquid, having red eyes, that's mostly mediated or to large part mediated by H1 receptors. Okay? The other role of H1 receptors is in the brain, where they seem to be involved in the regulation of arousal, of consciousness, so whether we fall asleep or not. We're going to talk a little bit more about that tomorrow when we talk about drugs that target this system. Okay? So H1 receptors, inflammation, allergy. H2 receptors have a completely different role. H2 receptors are found mostly on the cells of the gastric mucosa, of the gastric lining, of the stomach lining. Okay? And there, they regulate the production of acid, of hydrochloric acid. Okay? So the stimulation of this increases the production of hydrochloric acid in the stomach. Okay? If you have a question, just put up your hand and ask me. Yeah?
Yeah, you said the second role of H1 was stimulation of, or regulation of consciousness?
Yes.
So how can you explain the loss of consciousness? Is it a lack of histamine or?
No. Consciousness as a neurological phenomenon is extremely complex. Nobody really knows what's going on there, okay? But we know that certain neurotransmitter systems can either heighten consciousness or depress it, okay? And we know it mostly because the drugs that affect some of these receptors can do that, okay? So what I'm saying here that, you know, that noradrenaline or histaminergic system, adrenergic or histaminergic systems have a role there, that's because, I mean, we know that because some of the drugs that affect these systems also affect the level of consciousness. But you can't say that losing consciousness is caused by a lack of histamine. That it doesn't work like that. Okay? Yeah?
Can allergy affect focus?
No. Well, yes, in a way that, you know, it's distracting to you know. No, because histamine, as all the other neurotransmitters, does not cross the blood-brain barrier. Okay? So histamine that's produced elsewhere cannot get into the brain. Okay? So that's the explanation. Unless you had some kind of brain allergy, which I don't think you can get. Okay? So once again, H1 receptor connected to inflammation and all those effects of an allergic reaction. Well, not all of them. Many of the effects of allergic reaction. H2 receptors are responsible for regulating the production of acid in the stomach. Okay? Again, we'll talk more about those tomorrow when I tell you about the drugs and how they work and why these things happen that way. Okay? There are no questions about histamine. Yeah?
If they can't cross the blood-brain barrier, are there like mast cells or something that produce histamine in the brain? Because there isn't like a cerebral function, or like they cause sleepiness.
Yeah, actually, the histamine in the brain is produced by neurons. So they're histaminergic neurons.
Okay.
Yeah. Unlike in the periphery where they're produced in mast cells and other places. Yeah.
So H2 receptors are outside the brain? They're mainly...
That's a very good question. There might be some H2 receptors in the brain, possibly. I don't know. But again, clinically and pharmacologically, we're only interested. I mean, we know about H2 receptors in the stomach. It's possible that there may be some in the brain. Okay. The next neurotransmitter system we're going to be talking about is cholinergic. We're going to be talking about acetylcholine receptors. Cholinergic. So what kind of receptors for acetylcholine do we have? Does anyone know? Okay? Okay, so the basic division is between ion channels or ligand-gating ion channels and then metabotropic or G-protein coupled receptors. Okay? The first one, the first group, the ion channel one, they're called nicotinic acetylcholine receptors. Why do you think they're called nicotinic? Exactly, because nicotine is a specific agonist on nicotinic acetylcholine receptors. Okay? So both groups of receptors bind acetylcholine, but nicotine will only bind to nicotinic acetylcholine receptors. Okay? The other group, the metabotropic, the G-protein coupled receptors, are called muscarinic, which is once again after the specific agonist, muscarine. Has anyone heard of muscarine before?
In heart, on heart cells.
Okay.
Muscarinic receptors.
There are indeed muscarinic receptors in the heart, and we're going to mention some of them.
Decreases the heart rate.
They do decrease the heart rate, that's correct. But what is muscarine? It's one of the toxic substances in toadstools, in the mushrooms. You know, the mushrooms with the red cap? Yeah? Okay. Flying agaric, they also call it. Okay, so muscarine is one of the substances there. Okay, just one of them. The least interesting maybe, but anyway. So muscarine is a specific agonist on muscarinic receptors. There are further two subtypes of nicotinic acetylcholine receptors, but I'll deal with them together because there are no huge functional or biochemical differences between the two. So I'll deal with them together. Nicotinic acetylcholine receptors, as you correctly said, are ion channels, are actually cation channels. So mostly they let in sodium, okay, a little bit of calcium, maybe a little bit of potassium out. They're cationic ion channels, ligand-gated. And we can find them in the brain, we can find them in peripheral nerves, and we can find them in the neuromuscular junction. Okay, neuromuscular junction, yeah, the synapse between nerves and muscles. Okay, a special kind of synapse that contains nicotinic acetylcholine receptors. Yeah?
I don't really get this nicotinic. Like, if nicotine is an agonist for that, what is acetylcholine then?
Okay, acetylcholine is the natural agonist, the one that we have in our body. Okay, nicotine is an external. It's a pharmacological agonist.
And it binds to it as well.
Yes. Okay, it's a drug that binds to specifically to nicotinic acetylcholine receptor. Acetylcholine binds to both groups. Nicotine only binds to nicotinic. Muscarine only binds to muscarinic. Okay? All right. Let's now talk about muscarinic receptors. There are, again, at least 5 subtypes of muscarinic receptors. But I'll just mention some more details about 3 of them. As I said, they are coupled with, with G proteins. And their expression and their function differs between these subtypes. M1 receptors are mostly found on neurons in the brain or elsewhere. Okay, so they're neuronal. M2 receptors are the ones that were already mentioned here. They are the ones in the heart, also elsewhere, but in the heart. where they decrease, their activation decreases the heart rate. Okay? And the mechanism for that is, and I think I might have mentioned it already, the GI protein decreases cAMP via the inhibition of adenylate cyclase. But here the main effect on decreasing the heart rate is not mediated by the alpha subunit, but by the beta-gamma subunits. Okay? And it's actually very often the case with GI protein that the beta-gamma dimer actually causes most of the, or at least big part of the effect. Okay? So here it's mediated by the beta-gamma dimer. Yep.
Why is it mediated by the beta-gamma?
Why? Because that's how it is. The beta-gamma dimer is capable of binding to potassium channels and open them, which inhibits the cell, which hyperpolarizes the cell, etc. Okay? So it's just kind of an interesting thing that mostly what you see with G proteins is the alpha subunit that does all the interesting stuff. Okay? With GI proteins, and specifically here in the M2 muscarinic receptor, it's the beta-gamma that actually does most of the interesting stuff. Okay? The M3, the other muscarinic protein, the other muscarinic receptor, is found in exocrine glands. Okay? Do you know any exocrine glands? Pancreas? Salivary glands, okay, etc. Tear glands, okay? So these are regulated by acetylcholine, mostly via M3 receptor. Okay? So acetylcholine, first of all, is important in nerve-to-nerve transmission, okay, in the brain, elsewhere. In nerve-to-muscle transmission, where it causes muscle contraction, skeletal muscle, causes contraction. The muscarinic receptors, M1, again, is for communication between neurons. M2 mediates the effect of the nervous system onto the heart, decreasing the heart rate. M3 regulates glandular production in exocrine glands, again, via nervous system. Yeah? Yep.
What does M1 do again?
It's a neuronal one, so it's for communication between neurons. Both nicotinic and muscarinic receptors are found in the brain. And there, they have several different effects. But one of them, which is clinically relevant and pharmacologically relevant, is their function in memory formation. Okay? So the cholinergic system in the brain, again, the cholinergic cells are mostly found here in a small nucleus. It's not very important what it's called. Relatively small nucleus, but the effect is very important. Their projections into the cortex are used to conserve memories, to form memories. Acetylcholine, the cholinergic system in the brain, is damaged in Alzheimer's. Okay? So that's the cholinergic system. Do you have any questions? Yep.
You said at the beginning about nicotinic receptors?
Yep.
They're found in the brain, peripheral nerves, and something else.
The neuromuscular junction. Okay? So the synapse between nerves and skeletal muscle. Again, these things will be a little bit more, they will make a little bit more sense after we talk about the drugs, what the drugs do when they bind to these receptors. Because then I think it will all fall into place, why we need to know all these different receptors. Yep?
These cholinergic receptors with the Alzheimer's disease, they correlate with the kinase?
With?
With the kinase?
Why would it?
Because it doesn't, it's GQ pathway release calcium, and I thought this kinase is important in memory.
Yeah. The process of memory formation itself is so complicated that I don't really want to talk about, the specific pathways, because there are so many processes that go in there. It's quite possible that MAP kinase is involved in there, because even through nicotinic receptors, there is an increase in calcium. There are, let's not go there, okay? It's extremely complicated, yeah.
The structure goes up, both of them are cholinergic, but one is nicotinic, and the other one is
Muscarinic, yeah. So your question is that they're...
I was about to confuse about the structure of your nuts.
Yeah, both are acetylcholine receptors, okay? Nicotinic acetylcholine receptors are ion channels. Muscarinic acetylcholine receptors are G-protein coupled receptors, okay? Right, yep.
I think it's a general question about drugs. If nicotine is binding to the nicotinic receptors, does it mean that it compete with the acetylcholine, or?
Yes, it is a competitive.
So
Yeah, well, yeah, it's a competitive agonist. Yeah.
So does it have an effect that, that acetylcholine can't bind that much and then something happens?
Not really, they're both agonists. So nicotine will activate the receptor, acetylcholine will activate the receptor as well.
So there are enough receptors for everybody?
There are enough receptors for everybody. Well, I guess there are. I mean, nicotine will come, when you smoke or something, nicotine will come, bind to receptors that normally would not have any acetylcholine around, because the brain doesn't feel like it should be sending acetylcholine there. But the nicotine, because it just dissolves around, it will get there and activate them. So that's what causes the pharmacological effect. Okay. Right, we need to, move on, but yeah.
I just have a question regarding the muscarine. Is that something we naturally produce, or does it only exist in the toads?
Yeah, it's only present in the mushrooms.
Okay.
Yeah.
Okay. Yeah, that was what I was.
Yeah, it's a pharmacological. It's a pharmacological agonist. Yeah. We don't have it in our body. No. Right. Moving on. We finally move to the two most abundant neurotransmitters, so glutamate and GABA. We start with glutamate. Similar to acetylcholine, we have two groups of receptors for glutamate. One group are ionotropic, so ion channels, and the other group are metabotropic, so G-protein coupled. Okay, do you know these terms? Ionotropic, metabotropic? No? Okay, ionotropic means doing something with ions. Metabotropic means doing something with metabolism, meaning there is some signal cascade that involves enzymes and some metabolic changes. So that's the general division between, of receptors, the ionotropic receptors, metabotropic. The ionotropic receptors for glutamate. Any question? No? The ionotropic receptors for glutamate are further subdivided in a similar way as we had with nicotinic and muscarinic, so based on specific agonists. Okay, nicotine specific agonist and nicotinic muscarine specific agonist on muscarinic. Here we divide the ionotropic. They're named after their specific agonists, and their names are Nmda, which stands for N-methyl D-aspartate. That's the artificial pharmacological agonist that specifically activates NMDA receptors. Okay, only those receptors, nothing else. Now you'll see that it's a funny amino acid because normal aspartate that we have in our proteins is L-aspartate. Okay, this is D-aspartate, so a weird amino acid, and it has N-methyl on it. Yeah, it's just an artificial agonist. But that's what these receptors are called because they specifically respond to this artificial one. The other group are called AMPA. Here I won't even write the name of the agonist. It's quite significantly longer than that. And finally, the final group are kinase receptors, which are called after kinic acid. Again, an acid found, I think it's some marine organism that has this kinic acid, which binds to kinase receptors. Okay? So ionotropic glutamatergic receptors are NMDA, AMPA, and kinase receptors. Okay? They're all ion channels, they're all cation channels. Okay? Let me just say a little bit more about this NMDA receptor, which is extremely important for neurophysiology, and I'll explain why it's important. It actually plays an important role in memory formation, so you'll now hear a little bit about the system, how it works in memory formation. The NMDA receptor has two special properties. First of all, it is a cation channel as all the other ones, but it actually allows a lot of calcium in. Okay? All the cation channels, most of the cation channels will allow some calcium in, but NMDA receptor allows a ton of calcium in. Okay? And by allowing calcium in, it obviously causes all sorts of other processes in the cell. The other interesting property of NMDA receptors is that they need two inputs in order to be activated. All the other receptors that we talked about only need to bind their agonist and they open or activate a G protein or something. Only one input is needed. With NMDA, we need two inputs. One is binding of glutamate. Well, it's a glutamate receptor. But the other input, no, that would be interesting, but no, it's not GABA. The other input is membrane depolarization. So NMDA receptors will only open if glutamate is in the synapse, released from somewhere, and at the same time, the postsynaptic membrane is already depolarized by some other neurotransmitter. Does this make sense? Now, how does it work? Why is it interesting? Why do I, waste 3 minutes of lecture talking about this mechanism? Well, we can, in a very simplified way, we can imagine memory formation as basically integrating two inputs and joining them forever, permanently, in one output. Okay? When we touch a stove and we know that it's hot, okay, we need to connect stove, hot. Yeah, that's the memory we remember, okay, we shouldn't be touching, okay, in an extremely simplified way, okay? So imagine there is a neuron in the brain that corresponds to stove, and there is a neuron in the brain that corresponds to hot. And we need to connect the firing of those two neurons in an output that says, take your hand away or something like that, okay? So imagine we have the stove neuron firing all the time and releasing some neurotransmitter onto this new neuron, the memory neuron, okay? So the stove neuron is firing, but if it's just firing, well, nothing much happens. But if at the same time the hot neuron starts firing, these two inputs, open NMDA receptors, cause some further unspecified processes in the memory neuron, which then remembers that these two neurons, these two inputs, belong together. Okay? Extremely simplified way of looking at it. But I'm just using it to explain why this peculiar property of NMDA receptors. Why is it important? Why is it so important that neuroscientists really love this receptor? Receptor. They call it. There's a technical term for it. Coincidence detector. Okay, it detects coincidence of two inputs or several inputs. Okay, so NMDA receptor is called coincidence detector because it needs two inputs. It needs membrane depolarization and the release of, or the binding of glutamate. Okay, I won't say anything else about these two other ones other than their names. But then, since we talked about ionotropic glutamate receptors, we also need to say something about the metabotropic. I'll probably write the term down. There are many different subtypes of metabotropic glutamate receptors. They are usually abbreviated like this, mGlu-R, metabotropic glutamate receptors. So that's how we recognize them. If you see mGlu-R 1.5 or something, it's a subtype of metabotropic glutamate receptors. Their roles are many in the brain and elsewhere, but I'll just mention one which might make it easier maybe for you to remember that there exist, metabotropic glutamate receptor. Have you heard of the fifth taste, umami, the taste of glutamate? Yeah? Oh, the taste of meat, some people call it, okay? Yeah? There are 4 basic tastes, but there's also a 5th basic taste called umami. It's Japanese word. Umami? But that's actually the technical name for it, okay, even though it's from Japanese. So the receptor for this umami taste, which is meant to be a taste of meat or meat product or something like that, is mainly mediated by glutamate, and the sensation is mediated by metabotropic glutamatergic receptors. So in the tongue, we have these mGlu-Rs that detect glutamate or some other amino acids and mediate this 5th taste. Okay? Right, we're slowly running out of time. Well, quickly actually, running out of time. So let me talk about Gaba receptors, and the other receptors will be in a handout that I'm gonna post on the web. Okay. There aren't that many left actually, but yeah. Huh? Okay. Do you have no other classes after that? Okay. Well, those of you who need to leave, we can leave. I mean, it's not going to take a lot of time. It's going to take 5 more minutes or something. Okay, it's not a big deal. Right, GABA receptors. Well, actually, I could have left this here. GABA receptors are also divided into ionotropic and metabotropic. But fortunately, we only have 2 subtypes. So we have one ionotropic and one metabotropic. The ionotropic receptor is called Gaba A. And the metabotropic is called, what do you think? Gaba B. The Gaba A receptor, which is an ion channel, what do you think is what kind of channel is it going to be? GABA being an inhibitory neurotransmitter. So we need an ion channel that, when opened, will inhibit the cell.
An influx of chloride into the cell or potassium out?
Very good. So either it could be a potassium channel that will allow the efflux of potassium from the cell and hyperpolarize the cell, or it could be a chloride channel that allows the influx of chloride into the cell, thus hyperpolarizing it. Well, it is a chloride channel. Okay? It's a channel for chloride. When it opens, chloride goes into the cell, makes the membrane potential more negative, which means that it inhibits the cell. Okay, it makes it more difficult for it to depolarize and to do anything. Yeah? Makes sense? Good. The Gaba Be receptor is a G protein coupled receptor, and as a proper inhibitory receptor, it's coupled with GI protein. Okay, for glutamate and for GABA, there is no need talking about localization because they are everywhere. Okay, as we said, more than 90%, maybe 98% of neurotransmission in the central nervous system is mediated by glutamate or GABA. So they're everywhere, and they probably do everything in the brain. Okay. But we will talk about some specific drugs that target GABA receptors and that have some interesting effects. The final two groups of neurotransmitter receptors, and that's going to be very brief. I'm just going to mention their names, and that's about it. Well, pretty much. Opioid receptors. Opioid receptors. So what are the natural ligands for opioid receptors? Endorphins and dynorphins and enkephalins, so opioid peptides. Okay? Obviously, the artificial ligands for that are morphine, heroin, opiates. Also, yeah. There are three kinds of opioid receptors. Okay? And we're going to see a completely different naming convention here. They're called, they're labeled by Greek letters. Okay? And we have mu, kappa, and delta receptors. That's a delta. Okay? Mu, kappa, and delta. They are all metabotropic. They're all G protein coupled with the GI protein. All I'm going to say about them now. And the final receptor that I want to talk about is the receptor for nitric oxide, and you probably already know that. So what is the receptor for nitric oxide? No?
Isn't it a GS coupled receptor?
It's a?
GS coupled receptor?
Nope. It's not a G protein coupled receptor. Yes. It's a guanylate cyclase. It's a guanylate cyclase, and specifically it's called a soluble guanylate cyclase. It's called soluble guanylate cyclase because it's just dissolved in the cytoplasm. Okay? It just floats around the cytoplasm. It is not a membrane receptor. Nitric oxide is such a small molecule, it doesn't need to have membrane receptors. It can go into the cell very easily. So soluble guanylate cyclase is just an enzyme that floats around the cytoplasm, but when it binds nitric oxide, it becomes activated and starts turning GTP to cGMP. Okay? How do we terminate the signal here? So this is the activation of the signal. How do we terminate it? How do we stop the signaling? Okay, so there is a specific phosphodiesterase for cGMP. Okay, I'm just mentioning it because tomorrow we're going to talk about drugs that inhibit this phosphodiesterase. Do you have any questions? Just one more minute for questions. Yeah.
The AMPA.
Yeah. What they do. Again, as I said with GABA, glutamate is responsible for most neurotransmission in the brain. So they do everything. I picked Nmda receptor because it has interesting properties, and we sort of know why it needs these properties. But the other receptors are just everywhere, and they mediate all sorts of things.
And it's the same with the
Yes. Yeah. Okay. Any other question? Just one more minute, because maybe you'll be interested in the answer to this question. Maybe. Yeah.
So the chloride channel is actually like it polarizes again in the.
Yeah, I mean the chloride channel, the Gaba A channel.
Stops the potential.
When it opens, it allows chloride in, so the membrane potential becomes more negative. And if the membrane potential is more negative, the cell is hyperpolarized. It needs more inputs in order to be activated. So it's kind of pre-inhibited in a way, if you know what I mean.
So it's stopping the action potential.
It makes it harder for the cell to start an action potential. Okay. Okay, there are no more questions. That's it.